The immune system is constantly engaged in an internal battle to recognize and destroy disease-promoting agents within our bodies. It also develops an adaptive response, which, if an agent reappears, the immune system is primed and ready to again eliminate it from the body. Much of our knowledge of the immune system has come through research focused on its ability to eradicate invading pathogens such as viruses and bacteria. This includes the ability of the immune system to differentiate between self-antigens, naturally occurring tissues within the body, and the disease-causing invaders, which expresses foreign antigens. Activation of T cells and their interaction with target cells can result in the release of proteins such as perforin and granzymes into the cytolytic synapse. The perforin molecules are then thought to assemble to form a pore in the surface of the target cells. This acts to destabilize the cytoplasm of the target cell, as well as act as an entry point for granzymes. Granzymes are serine proteases that can trigger an apoptotic signaling cascade which causes the death of the target cell. Scientists now know that the immune system can also detect tumor cells. Although these cells normally evolve from cells already present within our bodies, they express antigens that the immune system can recognize and target. Such antigens can be presented on the surface of tumor cells, bound within a major histocompatibility class 1 or MHC1. This complex can then be recognized by T cells, which can initiate the destruction of the tumor cell. Tumor antigens can also be released from tumor cells undergoing apoptosis. Such antigens can then be ingested by dendritic cells, where they are processed and presented within an MHC1 and/or MHC2 to T cells. T cells interact with MHCs via T cell receptors on their surface, the activation of which stimulates cells of the immune system to release granzymes and perforin, which trigger apoptotic signaling cascades to initiate the destruction of tumor cells expressing the antigen. The killing of cancer cells by the immune system can lead to the release of further tumor antigens, which can then further enhance the immune reaction against these malignant cells. With time, cancer cells can evolve. Mutations within their DNA can generate a selective advantage, which can include mechanisms to evade detection by the immune system. This can include the loss of MHC1 on the surface of the tumor cell, reducing the ability of T cells to detect tumor antigens. Tumor cells can also overexpress inhibitory ligands and receptors which normally regulate T cell responses and can become an immune resistance mechanism. For example, increased CTLA-4 expression negatively regulates the early stages of T cell activation by down-modulating helper T cell activity and enhancing the immunosuppressive activity of T regulatory cells, or Tregs. Tregs suppress many effective arms of the immune response, and their presence is increased in many cancers. Similarly, tumor cells can also hijack the programmed death, PD-1 immune checkpoint pathway, to suppress immune control and evade immunosurveillance. PD-1 inhibits T cell function by engaging with its ligands, PD-L1 and PD-L2, the expression of which is increased on some tumor cell surfaces. Engagement results in the inhibition of kinases involved in T cell activation, acting as a brake on immune cell activation. The tumor cell or the tumor stroma cells may also release cytokines, such as TGF-beta, to generate a local immunosuppressive environment which promotes tumor survival. Tumor cells under selection pressure have also been shown to express indoleamine 2,3-dioxygenase, or IDO, an enzyme with immunosuppressive properties. The presence of IDO has been correlated with an increased proportion in the presence of a subset of T cells known as T regulatory cells, or Tregs. Tregs are potent immunosuppressive cells whose presence is elevated in many patients with certain forms of cancer. These examples highlight just some of the strategies employed by tumor cells to evade immune detection and illustrates the complexity that exists within the immune ecosystem. Amgen Oncology is looking for ways to harness the ability of the immune system to detect and target tumor cells. This includes the development and investigation of bispecific T-cell engagers, or BiTE antibodies. This technology utilizes the variable domains of two monoclonal antibodies. One of these two domains is specifically designed to bind to an antigen found on the surface of a tumor cell, whereas the other is designed to bind to CD3 on the surface of T cells. Investigational BiTE molecules are designed to act like a bridge between a tumor cell and a T cell. Amgen is also looking into ways in which viruses can be used to bioengineer T cells outside the body so they can recognize cell surface tumor antigens once delivered back into the body. These T cells can be modified to express chimeric antigen receptors on their cell surface to promote the engagement of the T cell with a tumor cell. T cells can also be engineered to express T cell receptors on their surface, which can enhance the recognition of tumor antigens presented within MHCs on the surface of tumor cells. Amgen Oncology is studying how modulation of glucocorticoid-induced TNF-related protein, GITR, could be used to enhance the immune response against tumor cells. GITR is activated by GITR ligand, GITR-L, expressed on the surface of antigen-presenting cells, such as dendritic cells. GITR ligation functions as a co-activating signal, driving TCR-induced T-cell proliferation, cytokine production, and activation to help develop anti-tumor immune responses. GITR is also constitutively expressed on the surface of Tregs, cells which suppress immune responses toward tumor cells. GITR activation can abrogate the suppressive activity of Tregs and/or increase the resistance of effector T cells to suppression by Tregs to enhance anti-tumor immune responses. This highlights some of the innovative approaches being evaluated by Amgen Oncology to harness the immune system to target cancer. The clinical effectiveness of these approaches is currently being investigated.